This lesson we're talking about DNA replication, and it's basically for SBI4U in Ontario. When we discuss DNA replication, what we're usually looking at is prokaryotic, okay? Because the eukaryotic version is a little more complicated, but we're just going to stick with the prokaryotic. It's much easier to look at. And when we look at replication, this is usually in the S phase of interphase. And usually we go through replication in order to do cell division. So we'll go through mitosis or meiosis after the cell has, or after the DNA is replicated. So remember, it's in the S phase where this is taking place. And when we look at it, you have to remember that DNA is a pretty massive molecule made up of many, many base pairs. And so you have to remember that all of this does take time. And remember, DNA, you have bases A, G, C, T, and A joins with T, G joins with C, C joins with G. I'm going to put 3 in there because it's actually 3, and T goes with A. So whenever you're looking at the base pairs, remember that they run this way, and remember they are an anti-parallel meaning, so they run up and down backwards. And remember, each of the bases is a nucleotide, and a nucleotide is made up of a pentose, or a 5-carbon sugar, a phosphate group, at least one, and a nitrogen-containing base. And remember, that's either a purine or a pyrimidine. So when you're looking at that, that's what you want to remember. When we continue with this, remember that at the replication fork in prokaryotes, you're replicating approximately 1,000 nucleotide per second. Okay, so when you're trying to figure out how fast this is going, DNA replication in a prokaryote runs at about 1,000 nucleotides per second being synthesized. So as we go to I pulled this image from Arms and Camp 4th edition, and you can see the structure of the DNA and how it's built. So if you look over here, you will see your sugar, your phosphate, and adenine here is your nitrogenous base. And over on the other side, you have thymine, which is hydrogen bonded to the adenine, and then the sugar and the phosphate. Note either end. This is the 3' end here of the sugar, and the reason being is the carbon 3, or the third carbon, is free at the end, whereas on the opposite side you will notice that the phosphate, which is attached to the 5 carbon, is free. Okay, so that's why it's the 5' end, and again they're running anti-parallel with your hydrogen bonds in the middle. What is DNA replication anyway? So let's go to the next slide. DNA replication. The thing about it is you need to start with your original strand, okay? And the original strand we will call the template, okay? And the template strand, again, is just a whole bunch of nucleotides stuck together. If it's a prokaryotic organism, it's going to be circular. If it's a eukaryote, it's going to be a long strand. I'm just going to write it out as if it was a long strand. And the enzymes that we're going to be dealing with here, there's quite a few of them. I'm just going to run through what it looks like as initially. So the first step that has to happen is you need to break open your DNA. Okay? And so you've broken it open, and that is done with a particular enzyme right here. And we'll have that as the red enzyme. Now hopefully I can remember the enzyme colors. Okay? And then it's got open, and then we will add SSBs, which are single-stranded binding proteins that will hold it from falling back apart. Now that the strands are open, you now have exposed bases that you can start building the new strand from. Okay? And so what happens before, it's like painting a wall. Before you can do anything, you need to prime it. So here, we're going to put some primers on. And the primer is basically just a sequence of RNA that adds to the end. And you'll notice that I'm doing it just from the bottom strand right now. And when we add the RNA primer, primase has done that, it's another enzyme. And from there, we're going to start another enzyme, and I'll put that as the triangle to start, and that triangle is going to start building from there and start synthesizing the new DNA as it goes along. Does that make a little bit of sense? So again, you started with a primer, the green enzymes attached, and started reading and building the new sequences along. On the other side, it would be nice if it could read in both directions, but these enzymes only read in one direction. So what you have to do is you have to put the primer downstream, and then the enzyme can stand there, and then we build in the other direction. And the same thing happens again a little further downstream. You get the primer, you add the enzyme, and you build. Okay? And so at the end of it, when you look, you're going to get the template strand, which is the blue one. And then when you started, you had primers and primers and primers in the top strand. Okay? And again, we'll go back to the 5' to 3'. And then we're going to have sequences of black, 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 and black. But notice the yellow is RNA. We don't want RNA, we want DNA. And then the other strand below, there's your template. Again, it's your 3' and your 5'. And then the yellow will be here. And then this one will just continue along until the end. Take a look at that and see what the difference is. I'm just going to put the little hydrogen bonds in there. Now just look at it and see what it looks like. Now as we look, the original strand is the blue strand, the black strand is the new strand, and the RNA, the yellow, needs to be removed. So another enzyme has to come in, and that other enzyme will come in, remove the yellow, and replace it with black. Okay, and that'll happen in different spots. Oops, and go black. And then we start getting DNA being formed in those little sequences. It doesn't actually happen here, but we'll get back to that in a second. Now you've got little fragments of DNA. What has to happen now is we need to glue it. So there's another enzyme that glues it here. So they glue them together, and then you get a sequence of one long strand of DNA, which is now your complementary strand, or your complement. Okay, so you have your complement, your template, and basically your DNA replication is done. You started with 2 strands at the top, which is it's one double strand, and down at the bottom after it finishes you have 2 new strands of the original. This is called semi-conservative replication. And semi-conservative just means that half the original stays with the new copy. Okay, let's move on. We'll just take a look at the next, and it's showing a very different visual. And again, this is from Arms and Camp, 4th edition. And this is showing, when you see the yellow, is the template or the original. Okay? The red is the complement that's been formed. And you will notice along the way that right at the yellow, right here and here, is your replication origin. It didn't start at the end, it started in the middle of the strand. Okay? So what happened was they split open the DNA, and then the helicase continued in either direction, but we'll explain that in a second. Notice the 3' to 5' is really important. It's always important, and you shouldn't forget that. Let's just do it in another format, and hopefully. And notice here the term Okazaki fragments. Well, the Okazaki, I'll start again at the end, and I just want to make sure we have the right colors. Again, we're going to start like this, and then we get there. Okay, and the first enzyme you want to look at, that red one that was ripping it open, the red is DNA helicase. Okay, again, remember enzyme ends in A's, helix, oh, it's the DNA helicase, it's ripping open the helix. Okay, so it's ripping open that. And then we added the little green dots, which were SSBs, which are your single-stranded binding proteins, and they prevent the DNA from collapsing back on themselves. The next step there was we added the yellow, which were your RNA primers. And they're just simple little strands of RNA that are added at certain areas, and they are there for the purpose of allowing DNA replication to happen. Okay? And again, I've marked those origins. And then the green, that enzyme that attached, the first enzyme that attaches is DNA polymerase 3. Okay, so the first one to attach is DNA polymerase 3, and the key there is it can read the template 3' to 5', and when it attaches the new nucleotides, it attaches by building in a 5' to 3' way. Okay, so what's going to happen is, as DNA polymerase starts to read through, it's going to start attaching these new nucleotides, forming the new DNA strand. Okay, and because it can only read 3 to 5, the bottom strand there is going to move really quickly, and that's where you get what's called the leading strand. Above, which I'm just going to draw down here, you have your 5', and then it ends in 3'. Again, DNA polymerase 3 cannot read 5 to 3, so the primer has to be inserted further downstream. DNA polymerase will attach, DNA polymerase 3, and it will build the new sequence in that direction. Okay? Again, then it jumps ahead, and we read and build again. And because that's happening, it takes longer, so that strand, as it's being built, is called the lagging strand. Okay? And then we're getting there. We're almost done. And I'm just going to flip to the next slide. A few slides. Japanese scientists small segments in the lagging strand and named them as Okazaki fragments. So that's basically DNA replication. The only that, again, that 5' to 3', I'm just doing the top strand right now. Because there's the primers there, what has to happen is we have to remove those, and the removal of the primer happens by DNA polymerase 1. Now, people are trying to remember, like, DNA polymerase 1, why wouldn't that be the first one to do? Don't go with that. Think of DNA polymerase 1 as smaller, so it's going to take the smaller bits out. And those smaller bits out, as they get removed, the yellow gets removed. So I'm going to take the yellow out. That's what DNA polymerase 1 does. And then it builds the new sequence here. Okay? Again, we'll do it the next one. DNA polymerase 1 moves in and inserts its new segment there. Notice there's still a few gaps here. They have not synthesized through your formation of the covalent bonds there through dehydration synthesis. So what you have to do is you need to add a new enzyme called DNA ligase. And DNA ligase is the glue that links it all together. And now you have two new sequences that are pretty much identical to the original. And that's DNA replication in a nutshell. The only thing I want to repeat is if you go, who discovered the lagging strand created these. Here is the presence of what's called telomeres. Okay? And telomeres are the sequences of DNA. If you looked at that 3' strand, remember there was the 3' to 5', and then what came on was a primer, and then the new DNA strand happened. Well, this sequence here, because it's at the end of the DNA, it does not actually get replicated. There is no enzyme that takes that, removes it, and adds new DNA. So each time DNA replicates, this sequence of DNA actually gets shorter. And so every time your cell undergoes synthesis of DNA or DNA replication, the DNA is going to get slightly shorter. And the region, there's a bunch of repeats down here that we call telomeres, and the telomeres will continue to keep getting cut shorter and shorter until there's a point when there's no more telomeres. And when there's a point where there's no more telomeres, you cannot replicate that cell anymore. So that cell will no longer divide. So think about cells in your body that no longer divide. They may have reached the end of their telomeres. And hopefully that makes a lot of sense. And please feel free to comment or let me know in class how this lesson was for you. Thank you so much.